Chromosomes clash day and night. Genetics laid bare under light. Bunsen burners flicker bright, all in the name of Mr. Right. Doing science right tonight. Answers never out of sight. Biology battles fierce and bright. Learning hard and feeling right.
Hi Year 10s, we're looking at the third PowerPoint in our Year 10 course. This is looking at chromosomes. Now I know we have looked at chromosomes a little bit in the last few weeks in our other videos, but we are just going to look at that in a little bit more detail in this topic. So going straight forward, as the other videos have all had, this is what you should know at the end of the week. So these are the 9 checkpoints that I'd like you to be ticking off at the end of this PowerPoint. Yes, I can do this and I'm ready for a test or an exam on this topic. So, moving straight into those, I'm not going to go through them in details, we'll just go through them for the slides. Going straight into them, we're looking at chromosomes in bacteria or prokaryotic cells. So bacteria themselves, they're types of prokaryotic organisms. Remembering prokaryotic from year 8 is a unicellular organism, doesn't have organelles, so it doesn't have a nucleus. Okay. Because of that, in most prokaryotic cells, there is only one chromosome, and it's circular in shape. Now, this picture here, I'm actually referring to the blue one. This is the chromosome that's in a circular shape. Ignore the red one that is very much looking like a circle in this picture. But the blue one here, this is the chromosome that's in a circular shape. Okay, in this one here, this blue chromosome, this has all the genes that the cell needs for basic life processes. So everything the cell needs to survive is in this blue circular chromosome here. Okay, the DNA is not associated with proteins, so we often describe the chromosomes as naked. And because there is only one chromosome, there's generally going to only be one copy of each gene. So you know how we talked about last week that humans had 23 pairs, so we have 2 copies of every gene. In prokaryotic cells, because there's only one chromosome, 1 copy of each gene. The only time you'll ever find 2 chromosomes in a bacterial cell or a prokaryotic cell is when cell division is happening and you've gone through DNA replication. Now this red section here, the one that actually looks like a circle, this is called a plasmid. Okay, so this is a DNA plasmid, and we're going to talk about that right here, but plasmids are small extra DNA molecules that are found in prokaryotic cells. Again, they're small, they're circular, and they're naked. They have a few genes that are beneficial to the cell, but they're not necessary for those life processes. Okay, so these things might include genes that help with antibiotic resistance, so helping them survive against antibiotics and create superbugs. These plasmids then are always going to be replicated at the same time as chromosomes of the prokaryotic cell through cell division and DNA replication. They're not going to be replicated at the same rate either. That means you could have a prokaryotic cell that's got multiple plasmids at once and technically may not be passed on through cell division because it may just stay in there. The interesting thing about plasmids too is they can be shared between different cells. So prokaryotic organisms can actually share those plasmids between them. Moving on to eukaryotic organisms. Now this is, if we talk about human chromosomes, this is going to be the general shape that you probably recognize, that X-shaped chromosome. But that's only when we're looking at DNA replication and chromosomes are duplicated. That's the only time you actually have that shape. This is much more likely what your chromosomes look like in your cells most of the time. In this case, so we're looking at eukaryotic organisms, it's a single long linear DNA molecule, and it's wrapped around a special type of protein called histones. So this is actually, if we zoomed in on it, we don't have that picture here. If you zoomed in on this, you'd actually see the DNA wrapped all around these little histones all throughout. Histones themselves, they are a globular protein, so spherical in shape, and they're actually wider than DNA, so the DNA just wraps around it consistently and coils around it. But what we're taking from here is eukaryotic chromosomes, long, linear, and wrapped around proteins, specifically histones. Okay, the differences between chromosomes. Now, to start with, chromosomes, they can only be examined during mitosis, because if we go back to eukaryotic cells here, these are actually too small and they're not condensed. when you're going through mitosis and you've gone through DNA replication, the chromosomes condense, so they become smaller, and they replicate. So there's 2 copies of the information there. So that's why there's that X shape there, because it's copied over. That means that there's more to physically see. And we normally label a chromosome as that X shape because that's how they were discovered, and we thought, oh, that's a chromosome. It's only later we discovered, hey, they're actually replicated information. But so this, when they're in this state, they're much easier to recognize. And it just so happens that's during mitosis, a type of cell division. And when we examine those chromosomes through mitosis, we can see that there's different types. And by types, we mean that the chromosomes differ in length because they have different number of genes on them. We have at least 2 types of every chromosome in every eukaryote, most species in more. Like we said in humans last week, there's 23 chromosomes in humans, 23 different types. Okay. Every type of chromosome that we have, so for example in humans, the 23 that we have, all 23 have different genes to each other. That means we need all 23 types to have all the genetic information to actually survive. Leading into a little bit more of a complicated topic is the phrase homologous chromosomes. Now, homologous means the same. So if 2 chromosomes have the same sequence of genes as each other in the same locations, we call them homologous. So for example, we have 23 pairs of chromosomes in our body, one from mum and one from dad, which means that we have homologous chromosomes. So we have 2 copies of chromosome 1, one from your father and one from your mother. They are the same chromosome, they have the same genes in the same location. There might be slight variations in the alleles of those genes, but they have the same genetic information. That means they are homologous chromosomes. They're not identical because of those alleles, but they are homologous. Okay. If we have 2 eukaryotes organisms of the same species, so 2 humans or 2 cats or 2 dogs or 2 fish, 2 lizards, we can expect that they will have the same chromosome types and that they will be homologous to each other. Okay, that means you can have interbreeding between members of the same species. So the fact that we have these homologous chromosomes between species, that means that members of the same species can actually breed together. Now, moving on that a little bit more is the phrase haploid and diploid. So a haploid nucleus, such as your eggs or sperm, they are known as haploids. They only have 1 chromosome of each type. So if we're talking about sperm and egg in humans, they only have 23 chromosomes total. They don't have 23 pairs, just 23 chromosomes. They're a haploid cell, 1 copy of each chromosome. Compared to our other cells, which we'll talk about in the next slide, are diploid cells. When you have those 23 cells, you're going to have 23 different types. So it's not a couple of homologous and a couple of non-homologous. It's you have 1 of each of the 23 types in humans. And that's a haploid cell. Okay, so haploid, 1 set of chromosomes. Compared to diploid, which has 2 copies of each chromosomes. So looking at all of our other cells, except of our sex cells, except our gametes, every other cell in the human body is a diploid cell. They have 2 full sets of chromosomes found within each cell. Now, the reason that we have our sex cells as haploid, so when the sperm and the egg meet and they both have 23 chromosomes, they form together to form a zygote, and that zygote then will have 23 from mum, 23 from dad, which makes 46 chromosomes, 23 pairs. If we just did that with diploids, then every time a new child was born, if we had 46 plus 46 and we go to 92 chromosomes, there's a problem. So in order to keep humans at having 46 chromosomes or 23 pairs of chromosomes, our sex cells, our sperm and our egg, have to go through a process called meiosis to become haploid cells. Okay. Moving on from that and looking at chromosome numbers. Okay, this is one of the key things that differentiates species between different types of species, is how many chromosomes they have. So if an organism has a different number of chromosomes, then they can't breed together. So for example, I'm taking the numbers off my top of my head, so the numbers could be slightly wrong. Humans have 46 chromosomes, and a lot of the great apes are only going to have 44 chromosomes. Okay. that means they've got 2 less chromosomes, but because they've got 2 less chromosomes, we can't breed together, because when you go to those haploid cells, you've got 22 and 23, they can't come together to form 23 pairs or 22 pairs, so that species can't breed together. Okay. Evolution is one of those processes that's going to help change the number of chromosomes an organism has. It can decrease because chromosomes confuse together, or it can increase because chromosomes split apart. And we'll watch a video of that when we talk about chromosome number. We might even watch that video in class this week, talking about evolution and chromosome numbers, and looking at how chimpanzee chromosomes differ from human chromosomes. We will actually watch that. I'll link that on Doing Science Right. That's a good video to watch for this. If we're looking at different types of species, so we've got a few different types of species here. Sorry, I was wrong. Chimpanzees actually have 2 extra, that's 48, not 44. All eukaryotes have at least 2 different types of chromosomes. That means their diploid number has to be at least 4, because they've got 2 different types of chromosomes. Therefore, when they've got 2 or a pair of each one, that becomes doubled. So we have 23 pairs of chromosomes, which means we have 46 chromosomes. A horse threadworm has 4 chromosomes. Rice has 24 chromosome diploid number. Chimpanzees 48, and dogs at 78. There are some eukaryotes that have a diploid number over 100, but this is just a nice way to compare those species. Chromosomes are also important, though. It's not just for everyday the genetics determining characteristics. It's also important for determining sex. So there's 2 chromosomes in humans that determine sex. We have the X chromosome and the Y chromosome. The X chromosome is relatively large, and all members of the species, male or female, they both have the X chromosome. The Y chromosome is much smaller. It's actually shrinking in size. It used to be the same size as the X chromosome. And that's only found in males. Okay. Now, because they determine sex, they're called the sex chromosomes. Okay, so we have 22 autosomes in our body. So they're the cells that are the chromosomes that aren't determining sex. And then we have the 2 sex chromosomes, X and Y. Okay. X chromosome is really important. It has lots of genetics or genes that are important to both males and females. Therefore, everyone has to have an X chromosome. You can sort of see here the X here. The female has homologous chromosomes, 2 X's, but the male has an X and a Y. Okay, and you can see here the Y is considerably shorter. Okay. Very small part of that Y chromosome has the same information as part of the X chromosome, but the rest is different, helping to determine if it's a male or a female. So specifically, when we're looking at gender, it's the Y chromosome that will cause a fetus to develop as a male. If you don't have that Y chromosome, you don't develop as a male. All children are going to get 1 X chromosome from their mother. So the mother, because it only has 2 X's, can only pass on an X. So you can see here the female child's got X from mum. Male child's got an X from mum. So therefore it's up to the male. If a male gives an X chromosome, it's going to be a female. If the male gives a Y chromosome, it's going to be a male. In terms of the Y chromosome getting smaller, it's because there is only 1 Y chromosome in the body. For the X chromosome, and for all our other chromosomes in our body that are homologous pairs, if there are problems or breakdowns of the chromosome, we have another copy of the chromosome that we ensure is passed on, or we can repair that chromosome from that information. But the Y chromosome is always by itself. It's never going to have 2 Y chromosomes unless it's a genetic disorder, which we may talk about a little bit later. But generally, there's not going to be 2 Y chromosomes in anyone's body. And that means that there's no information to fix the Y chromosome, or if there is problems, we can't pass on the good copy of the Y chromosome. And because of that, over time, the Y chromosome has consistently gotten shorter. Now, nowhere near in our lifetime, we're looking millions and millions of years, there is the possibility that the Y chromosome will completely disappear in humans. This has happened in other species of mammals, and those mammals have just found new ways of determining sex. For this is great, we've learned a lot about chromosomes today, but then if we're looking at how we look at these, and that's a karyogram. So this is a karyogram of human chromosomes. Over here, we are being able to see all of the chromosomes in an organism during mitosis. Okay, the clearest stage that we can actually see all of the stages, all the chromosomes, is actually during metaphase. And we use different stains to make those chromosomes show up. A cover slide is forced onto the microscope slide. That causes the cells to burst and the chromosomes are spread out across the slide. We use a microscope with no overlapping to find a cell that has no overlapping chromosomes, and we take pictures of them and arrange them digitally by size and structure to get this nice karyogram where we can see all of them here. So you can actually see here that we've got 2 chromosome 1, 2 chromosome 2, 2 chromosome 3, etc., and then chromosome X and Y. All up, we have 22 pairs of chromosomes plus our sex chromosomes. Okay, so 22 autosomes, 2 sex chromosomes. You can see all of these are homologous chromosomes, except for these ones down here, the sex chromosomes. We've got the X and the Y. If it was 2 X's, they technically would be homologous chromosomes as well. Lastly, looking at a karyogram for someone that has Down syndrome. And you may be able to ask to actually look at karyograms and test an exam and actually tell me if they have Down syndrome or if they're male or female. So straight away with this one, we can see there is 2 things here that can help point out some information. First off, we have homologous chromosomes at X. So we have 2 X chromosomes. This has to be a female. But the unusual thing about this karyogram is this person has 3 chromosomes at 21. And that is what Down syndrome is. Down syndrome is called trisomy 21, which means you have 3 chromosomes at the 21st chromosome spot. And that is what Down syndrome is, 3 of chromosome 21. Okay, scientists will use karyotyping to actually determine if someone has Down syndrome. And this is why techniques like this are really important to help diagnose conditions such as that. This is the end of the PowerPoint. I know there's a lot of information. We've jumped around a lot. We will go through this in class. I will put some supplementary videos down below this video as well. Let's discuss this in class. Bye.